I think this is like what sharing session for all of us. So I do hope that this presentation will benefit everyone. Thank you. Right. My topic will be Burkholderia pseudomallei, the causative agent of meliodosis. Right, for the presentation outlines, I will start with history of meliodosis, then followed by meliodosis cases, meliodosis, then treatment and prevention, diagnosis of meliodosis, then followed by Burkholderia pseudomallei and salt isolation, then summary. Right, we start with history of meliodosis before we go further, deeper into the topic. So meliodosis was first described in 1911 in General Hospital Rangoon, Burma by Sir Alfred Whitmore and his assistant Krish Naswamy among 31 morphine-addicted patients. Actually, they found among 38 patients, but 7 of them were not morphine-addicted patients. And then at the very beginning, they thought that the disease was caused by Burkholderia mallei, the causative agent of glanders. However, when they did a thorough investigation, they found that none of the patients had a history of recent animal exposure. So they start to dig more deeper about the disease. And then they found about meliodosis. So meliodosis is one of the infectious diseases caused by a gram-negative bacterium, which is Burkholderia pseudomallei. Meliodosis was also called Whitmore disease. This one was taken from Sir Alfred Whitmore name. And then followed by Vietnamese time bomb. This name they got during the World War I and World War II in Vietnam, because many of the US Army got infection of Burkholderia pseudomallei during that World War. And then followed with nightcliff gardeners disease, because many farmers and gardeners got infection, followed by morphia injector septicemia and paddy field disease. In 1921, Sir Stanton and Fletcher called the disease meliodosis, derived from a Greek word meaning glanders-like disease, a zoonotic disease of horses. Meliodosis mainly affects lower-income populations living in rural areas, and it is estimated to cause nearly 90,000 deaths a year globally. And these happen in endemic areas. Then distribution of meliodosis worldwide. Annual death of meliodosis worldwide among human population, including endemic meliodosis and disseminated meliodosis, is about more than 150,000 cases. Half of the cases were reported due to the inadequate treatment. So for the endemic meliodosis, it happened in Southeast Asia and Northern Australia regions. While disseminated meliodosis, the cases were reported from other countries, including in United States and area in Mississippi. And look at to this map, you can see very clear that the endemic area colored with red color, this one is Southeast Asia area and then Northern Australia. And the other part in the map were disseminated meliodosis. And the color belongs to, the color actually, it depends on the total number of meliodosis happened. And then the total number of the cases actually were taken from 1910 to 2022. Right, meliodosis cases. About endemic meliodosis, we start with meliodosis cases in Malaysia. Meliodosis was first reported and documented in an outbreak in 1913 involving an outbreak of meliodosis among laboratory guinea pigs at the Institute of Medical Research, Kuala Lumpur, now in NIH, Setia. Since then, meliodosis has been recognized as an endemic disease in Malaysia until now. Right, followed by the first case of meliodosis in Singapore, which was reported in 1920. An increase in the number of meliodosis cases happened in March and April during heavy rainfall, strong winds, and flash floods. And then in Indonesia, the first meliodosis cases happened in Java Island in 1929. And then in Thailand, indigenous meliodosis was first reported in 1955 with lymphadenopathy cases. And then the first case of human meliodosis in Vietnam was detected in 1925 in a female patient pregnant in the 5th month, living close to Saigon. Now it is called Ho Chi Minh City. 6 days after the onset of symptoms, the patient had a miscarriage, and after 14 days of illness, the patient died. So meliodosis was first reported in Australia, Northern Australia, in 1949, following an outbreak in sheep in Central West Queensland. And the first human case of meliodosis happened there was in 1950. And disseminated meliodosis or sporadic human cases was reported among US Army after they come back from the Second World War, where the primary infection happened in Vietnam. Then moving to the next slide, now we are talking about meliodosis. We start with disease at a glance. Right, the disease is caused by Burkholderia pseudomallei, a potential biological warfare, and then it is grouped under hazard group 3 by CDC, and it is highly infectious. Portal of entry is through digestion, inhalation, and inhalation is the most infectious route, and percutaneous. This one normally skin injury. Person-to-person transmission is rare. However, laboratory exposure is a risk. Burkholderia pseudomallei must be handled in a containment laboratory, which is in endemic area, it should be in a biosafety cabinet too. But for the endemic area, non-endemic area, they should use BSL-3, which is to reduce the risk of laboratory-acquired infection. Meliodosis predominantly occurs in people with frequent contact with contaminated soil or surface water in endemic areas, such as workers in rice paddies, particularly during the rainy seasons. Cases may increase after hurricanes and heavy rain because the bacteria rise to the soil surface. Travelers to rural areas in endemic countries may be at risk if they come into contact with contaminated soil or water, especially if they are immunosuppressed or have a chronic condition such as diabetes. So next slide, more about meliodosis. Right, drinking untreated or unchlorinated or unboiled water increases the risk of infection, especially in endemic area. The disease typically has clinical manifestations such as pneumonia, sepsis, and abscess formation. The disease is predominantly in persons with underlying risk factors, especially diabetes, excessive alcohol use, chronic lung disease, including COPD and cystic fibrosis, chronic renal disease, thalassemia and malignancy, or other non-HIV-related immune suppression. In healthy people, exposure to Burkholderia pseudomallei may be associated with asymptomatic seroconversion, whereby the time period where the antibodies develop and become detectable in the blood. Look at to the pictures on the right side. You can see that like carbuncle. This is skin infection, localized infection happened among meliodosis patient, and this one is pulmonary infection, pneumonia infection, and this one also skin infection. This one lymphatic infection. Right? And meliodosis presents in 4 general clinical forms: localized infection, including ulcer, nodule, or skin abscesses; pulmonary infection, bloodstream infection or septicemic; or disseminated infection, where most of the cases involve many internal organs. Brain abscesses may be seen but less frequent. Latent reactivation or relapse infection can occur months to years after primary exposure, and this one based on the what happened among US Army when they came back from the World War in Vietnam. Meliodosis can be life-threatening or fatal if left untreated. Complications of meliodosis include sepsis, organ failure, pneumothorax, cardiac tamponade, or fluid build-up in the pericardial area, and recurrence of infection. Other infectious diseases that are always suspected with meliodosis, especially tuberculosis or chronic malio, means that the pneumonia infection. Staphylococcal diseases with sepsis, invasive Klebsiella infection, cat scratch disease, glanders, plaque, and tularemia. You can see this is examples of the meliodosis infection, right? For clinical manifestation, there are 4 clinical manifestations of meliodosis. 1, localized infection, where with meliodosis results from direct inoculation through a skin abrasion. A nodule, ulcer, or abscess develops at the site of entry. It can progress to septicemia or bloodstream infection, osteomyelitis, meningitis, and brain, liver, or spleen abscesses. Then followed by pulmonary infection. This infection is the most common presentation for meliodosis. The lung infection can become chronic and may be mistaken for tuberculosis. And next, septicemia or acute bloodstream infection. This infection is the most severe form of meliodosis. It occurs mainly in people with underlying chronic conditions such as liver or kidney disease. The onset of septicemia is rapid and often leads to septic shock, and the mortality rate for septicemic meliodosis can exceed 80% out of the total cases. And disseminated meliodosis abscess develop in various body sites such as skin, lymph nodes, liver, and other internal organs. And then you can see on the left side, the area of the infection. Meliodosis, just like tuberculosis, it can infect any part of human body, including area head and neck, respiratory system, gastrointestinal system, skin and soft tissue, and musculoskeletal system. Right, signs and symptoms of meliodosis. The incubation period of the infection is generally 1 to 21 days, one week to 3 weeks, with a median of 4 days. People who receive high inoculum can become symptomatic within a few hours. This one normally when people, got a primary infection through digestion, especially just like what happened during the Lubuk U case last time in 2010. Just overnight, the patient died because of they drink a lot of water, contaminated water at Lubuk U. Right, symptoms of meliodosis usually develop within 1 to 4 weeks after exposure, but approximately about 9% of the patient got the symptoms even more than 2 months of exposure. So patients usually have high fever, swelling, muscle aches, cough, chest pain, headache, confusion, seizure, loss of appetite, and lost weight. Chronic meliodosis normally end up with a disseminated infection and causes problems with the liver, spleen, prostate, joints, bones, lymph nodes, skin, or brain. Right, on the right side are the other side of meliodosis infection. Meliodosis also can happen in central nervous system, cardiovascular system, urinary tract system, and other part of the human body. Right, for you to take home notes, right, bear in mind that risk factors for developing meliodosis. For clinical factors, people who have diabetes, excessive alcohol use, chronic lung disease, including chronic obstructive pulmonary disease or COPD, or cystic fibrosis, chronic renal disease, thalassemia, malignancy, or other non-HIV related and immune suppression, they are prone to get the infection much more than healthy people who have no clinical factors that I mentioned just now. And the activity factors that will fasten up the infection, including any activities that involving with direct contact with soil or water in endemic areas of meliodosis, and no precautions are taken. Drinking untreated or unboiled or contaminated water or food in endemic areas, and dealing with Burkholderia pseudomallei with improper personal protective equipment. This one for healthcare workers especially. Right, for treatment, there is currently no effective vaccine for meliodosis. Treatment of meliodosis requires long-term antibiotic therapy, acute phase followed by eradication phase. Currently, ceftazidime is the treatment of choice during the acute phase. This one more on IV. Antibiotics are given intravenously initially for the first 10 to 14 days. Oral treatment usually trimethoprim, sulfamethoxazole for 12 to 20 weeks. Continue with antibiotic treatment is required for several months to prevent relapse. Severe cases of meliodosis may require admission to an intensive care unit or ICU for supportive therapy, and abscesses may require surgical drainage. For prevention, the risk of exposure to Burkholderia pseudomallei can be minimized by avoiding contact with soil and standing water in endemic areas. This especially applies to people with skin wounds or chronic health conditions. Agricultural workers in endemic areas wearing rubber boots when working. And then appropriate use of personal protective measures or equipment by healthcare and laboratory staff. Label samples appropriately if they suspect meliodosis to protect lab personnel. And avoid uncooked and unboiled water. Moving to the next slide. And then this is the treatment regime that can be used as a reference. This one was taken from journal in 2016. Maybe they got some updates on the treatment, but still you can use as a reference for the meliodosis treatment. Okay. Okay, I won't go deeper about this one. Right, diagnosis of meliodosis. Okay, based on Sir Alfred Whitmore, diagnosis of meliodosis, close collaboration between the clinic and laboratory were essential to good quality of modern medicine. Right, two different type of diagnosis, one clinical examination and another one is laboratory diagnosis. In my presentation, we will focus more on laboratory diagnosis. The aim of laboratory diagnosis is to identify the causative agent of the diseases to assist the healthcare provider to choose the most effective treatment because certain antibiotics are more effective against specific bacteria. So in laboratory diagnosis, especially in microbiology lab, bacteriological investigation is always number one, right? Okay, culture is the first choice in a microbiology lab because it is a confirmative method. Culture is the gold standard method for confirming the diagnosis of meliodosis. A full screen should be sent for culture, including blood, sputum, urine, throat swab, tissue, and any aspirated pus. Selective media increase the yield from non-sterile sites. Okay, laboratories must be informed of the suspected diagnosis for two reasons. First, the organism may be dismissed as an environmental contaminant. And second, to ensure the use of appropriate media and laboratory containment when we are dealing with clinical specimens suspected with meliodosis infection. And besides the culture, we will use conventional methods. The conventional methods including biochemical assay, API20NE, latex agglutination, and microscopic observation based on gram stain. Right. And then besides the conventional method, we also use molecular methods, including PCR-based assays, qPCR, DNA microarray, ribotyping or fingerprinting, and RFLP. And actually, we do have more, but this is what we are currently use in our lab. And also in Thailand, they use these molecular methods. And for the automated one, we also use BACTEC, BioTech 1 or 2, Biotyper, Broker, Rapid NF+. We do sequencing, and the most recent one is Meliodosis mass spectrometry. And then besides the bacteriological investigation, we also do serological investigation because serological tests are often performed as preliminary tests in endemic areas to expedite the diagnosis. Also, we want to know the exposure to the meliodosis. So methods that normally we use for serological investigation, ELISA, which is for diagnosis and prognosis. Lateral flow, the latest one, diagnosis and prognosis. I had diagnosis in non-endemic area, but for screening or seroprevalence survey in endemic area. IFAD for diagnosis and prognosis. Immunochromatography, which is rapid IC, using IgG for diagnosis in endemic areas, but rapid IC IgM detection for diagnosis in non-endemic areas. Right, this one just option actually for the bacteriological investigation. This one I just share what I got from Konkan Hospital University in Thailand. When they deal with blood and pus, they use blood agar and McConkey. For optional, they inoculate 0.5 ml or 1 ml blood sample into BHI, brain heart infusion broth, and incubate for 4 hours at 37 degrees. Then they will sub the colony on BA and McConkey plates. And then they also use incubate. Incubate the sample in BACTEC machine. But this one just only in a big general hospital. After has a positive signal, they culture the sample on BA and MAC plates. For tissue, they use blood agar, McConkey, and CHOP agar. For sputum, they use blood agar, McConkey, gentamicin blood agar, and H2 media. While for urine, they use 1 microliter urine using calibrated wire loop and sub on blood agar first, then follow with another selective media. This one just optional. I just want to share information that I got, that I have learned in Konkan University Hospital in Thailand. Right, selective media for Burkholderia pseudomallei identification that we use and other endemic country use, including H2 agar. And then the H2 agar, you can see there are 2 different colors. Actually, this one based on the brand that you use, especially for crystal violet and a phenolate based solution. Different brand of crystal violet, sometimes they give different color a little bit of the H2. Sometimes you can see like red color, and then sometimes you can see like violet color plus red a little bit. Right, okay, so H2 agar was invented by Sir H2 in 1979. The media contains crystal violet and gentamicin to inhibit the growth of gram-positive and other Pseudomonas species. To isolate Burkholderia pseudomallei from non-sterile clinical specimens, this selective media is very powerful, is very useful, especially to isolate Burkholderia pseudomallei from non-sterile specimen and also environmental samples. So another selective media that that many lab are using it now, which is Burkholderia cepacia selective agar, BCSA. This one because, you know, sometimes people or lab technician misidentify between Burkholderia cepacia and Burkholderia pseudomallei when they use normal blood agar or McConkey. So because this media contain crystal violet to inhibit gram-positive, and they use vancomycin to inhibit enterococci, polymyxin and gentamicin to inhibit other aerobic and gram-negative, including Pseudomonas aeruginosa. And for Burkholderia pseudomallei, the morphology is totally different. On this plate, you can see is Burkholderia cepacia. So next media is Francis media. This one was invented by our pegawai sains, if I'm not mistaken, from Hospital Kuantan in previous time, which is Mr. Alec Francis. So this Francis media contains gentamicin and crystal violet. This media is to differentiate Burkholderia pseudomallei and Enterococcus and other Enterobacteriaceae. So for Burkholderia pseudomallei, the bacteria will grow, but with less color of the yellow one compared to the Enterobacteriaceae. Right. So this one is the Francis media. This figure, this picture actually I took from journal by Francis, Mr. Francis. where you can see Burkholderia pseudomallei, Burkholderia cepacia, and Pseudomonas. After 4 days of incubation, they show different morphology on the plate. For Burkholderia pseudomallei, you can see the greenish color one here, wrinkle, greenish, wrinkle, and a bit dry. Okay. For Burkholderia cepacia, you can see different color. The color is more on blue color and a bit mucoid. While for Pseudomonas, you can see pinpoint and small, like this one. Pinpoint and small. Right, for Columbia agar, this one for Columbia agar, normally we will not use this media for routine diagnostic in our microbiology lab, but we use this especially when we are dealing with environmental samples. We use Columbia agar. The media contains peptone mixture and yeast extract as a source of nitrogen, essential vitamins, and amino acids to isolate fastidious microorganisms, especially from environmental samples. And the colony that you see on this plate belongs to Burkholderia pseudomallei. You can see the wrinkle one. Okay. Next, chocolate agar, McConkey agar, and chromogenic agar. So McConkey agar, sometimes it's a bit tricky. Sometimes we identify E. coli and Burkholderia pseudomallei sometimes, because Burkholderia pseudomallei on McConkey, it should be non-lactose fermenter, which is colorless. But I don't know, based on my experience, sometimes I got similar color to E. coli. So, right. So now we're moving to the next slide. The topic that we will talk, that I will present is Burkholderia pseudomallei. So Burkholderia pseudomallei, it is an environmental pathogen, soil saprophyte, living in soil and water in endemic areas, primarily South Asia and Northern Australia. Previously, the species was called Bacterium with mori, Meliodosis pseudomallei, Lophrella pseudomallei, Piferella pseudomallei. In 1992, the species is classified in the Burkholderia genus based on its RNA properties, and it has been named as Burkholderia pseudomallei. The Burkholderia pseudomallei strain can be easily isolated in wet areas, especially in paddy fields, deforestation such as Felda and Vectra, and farming areas, including Rumah Hutan in Sarawak. Last time I was there for sample collection. The isolate can be found in clay layer or soil layer about 25 to 30 cm underneath the soil surface and distributed in the environment during the rainfall. It is intrinsically resistant to aminoglycosides, including gentamicin, amikacin, and streptomycin, and macrolides, including clarithromycin and erythromycin, due to efflux pump activities. So, bacteriology characteristic of Burkholderia pseudomallei, including it is gram-negative bacilli. The size is about 2 to 5 millimeter. It is motile, non-performing organism, sugar oxidizer. It is oxidized and catalyst. It has oxidase and catalyst positive, indole negative. It shows metallic sheen within 24 hours of incubation. It has bipolar staining under microscopic observation. It can grow at 42 degrees. It is pleomorphic, means that under microscope, you can see the bacteria, the pathogen, not only not only bacilli shape, but sometimes it can be in cocci shape. A mixture of cocci and bacilli we call pleomorphic. And then it has wrinkle colony, especially after 72 hours of incubation on most selective media, even on black agar and McConkey. And it is resistant to gentamicin, polymyxin B, and colistin. So moving to the next slide. This is some examples of Burkholderia pseudomallei, how it looks like under this one, under a light microscope after gram stain. Gram-negative bacilli, you can see a bipolar staining, not very clear, but bipolar staining, I think everybody knows that, especially from microbiology lab. And then even sometimes you can see something like spore at the both edge of the bacilli, but that one is not spores, right? So this one another figure of Burkholderia pseudomallei, and this one is SEM observation, scanning electron microscope picture, show gram-negative bacilli. Right. Okay, next. Okay, main reservoir of Burkholderia pseudomallei in endemic areas. So Burkholderia pseudomallei easily to be found or isolated from soil, and then in paddy area, in contaminated area, and then sometimes in not sometimes, but if in what rumah hutan, this one is rumah panjang or rumah hutan in Sarawak, and then from unboiled or untreated water. Right. Pathogenesis of Burkholderia pseudomallei. Burkholderia pseudomallei rapidly shift from an environmental organisms to an aggressive intracellular pathogen, and it is capable of rapidly spreading around the body. The expression of multiple virulence factors at every stage of intracellular infection allows for rapid progression of meliodosis infection. So actually overall, they have about 5 to 6 stages of infection before it cause disease. Start with the pathogen invasion into the human cells or host cells, and then after it is being phagocyte, and some of them will be get rid by our immune cell, and then some of them will stay in the phagosome, and then they can survive using their own metabolic systems, and then they start to escape. After they escape, they start to polymerize, propagate number of their colonies by manipulating the host cells, using energy from the host cells for their replication. And then they start to propagate more and more, and then until they start to spread to human or host cells, and then cause the disease. So the expression of multiple virulence factors by the pathogen at every stage of intracellular infection allows for rapid progression of infection. Several secreted virulence factors manipulate the host cells, utilize energy from the host cell for their replication. And then one of the very powerful secretion system that Burkholderia pseudomallei has is a type 6 secretion system, where this type 6 secretion system will facilitate the metabolic pathway in the host cell for cell-to-cell dissemination within host cells. Right. Next slide is about Burkholderia pseudomallei isolation from environmental samples and the identification. And this figure actually on the left side, I didn't wear a proper because this one after we finish collect sample, soil sample. This one was taken when I was in Konkan, Thailand. I followed the team for during the outbreak to isolate to do sample collections in their area, periphery area. And then on the right side was taken when I was in Bintulu, Sarawak for sample collection for sampling, soil sampling for Burkholderia pseudomallei as well. Right. So for soil sampling, what we did first is we dig a hole to the soil about 30 cm depth. Okay, we have to use a scoop, and that scoop, before we dig, we have to sterilize it using alcohol, and then we wipe, and then we start to dig, make the hole about 30 cm depth, and then we measure using ruler. And then next step, what we did, we collect soil sample in a sterile condition, means that we have to wear a proper PPE, especially glove. Okay, and then the soil should be collected in sterile condition where we have to use theraplastic bag when we want to get the soil, and then we seal it the soil in the sterile plastic bag. Right. So next step, what we did, we start to process the soil samples. What we did, we mixed the raw water with 100 gram soil sample, and then we leave it we leave the sample at room temperature for overnight. So this one I did in Khon Kaen University, Thailand. So this is how it looks like lah, the sample after you process, and then you dissolve in sterile water, and then you stand it at room temperature for overnight to make sure that all the bacteria, including Burkholderia pseudomallei, will go up to the water level, to the water surface, not inside the soil anymore. So next, what we did, we start to do plating. So we plate the specimen, the samples on the H-done media. So this is how it looks like lah. Right? We use a hockey stick. All right, okay. And then this is just some of the examples what we got after we do plating. So we will have so many type of colonies, different species on the plate. So this part will be a bit tricky because we have to very careful with all with every single of the colony or isolate that grow on the media. We cannot skip any of the colony. If you see 5 or 10 5 to 10 different colonies, you have to pick up, and then you start to resub on another plate, and then we have to incubate the plate up to 7 days. And then if you want to incubate it at incubator at 37 degree, you have to seal it because we don't want the plate dry very fast. And then if you do on bench, but actually it is not recommended, but in Khon Kaen, Thailand, because highly endemic of Burkholderia pseudomallei, so they just seal the plate and then put on the bench. All right, okay. So this is one of the example when you do plating at different concentration of the different dilution of the samples, you will have different actually number of colonies on the plate. This just one of the example, and then it is too much actually, this one, right? If you got like this, you have to further dilute your samples because it's very difficult to pick single colony. But this one actually just example. This is the one that I already isolate. This is the pure culture one. It's not the mixture like this one. Right. These two figures also examples of the plate with the grown colonies. This one actually H-the media, but different, but the color is slightly, like red or what, something like light red, because of the crystal violet that I use is not very good one. So you can get something like this, so many type of colonies. So another examples of the colonies from soil samples, right? Like this one also. This one, the pinkish one, Burkholderia pseudomallei. This one based on my investigation, right? So for the identification after you got the colony, the single colony, what we did before, we direct use latex agglutination test, because Khon Kaen, they are the one who produce latex agglutination kit. So we directly use a latex agglutination kit. We just pick one single colony and then add with latex agglutination, and then a few seconds you can see the results. So we follow with the commercial API 20NE biochemical testing strip by Maru. And below, common profile numbers for Burkholderia pseudomallei. You can use this if you use API 20NE. Right. So another biochemical test we use when we do identification for the Burkholderia pseudomallei isolate from environmental samples. Actually, it's similar to what we do for clinical isolate of Burkholderia pseudomallei. This one TSI result after 24 hours. This one on the left one Burkholderia pseudomallei, on the right one Burkholderia pseudomallei. TSI because Burkholderia pseudomallei you can't see any orangey color in the triple sugar iron. Okay, and then for BP, this one after 48 hours, you can see slightly orangey, but not really orange if you compare to Sipasha. Triple sugar iron, this one day 1 and day 2. This one isolate from from soil samples. This one for TSI. Right, next we did a citrate biochemical test. Citrate for Burkholderia pseudomallei sometimes from if the isolate from clinical samples, the citrate very fast. You can get positive very fast. But for isolate from environmental samples, sometimes it's a bit delay the result. It become weak citrate. Like this one, you can see the second tube. Even after 24 hours or 48 hours, sometimes you still see a green color. But slowly after 48 hours, you can see slowly the color on the top of the slope change to blue color. But it will take time. It's very it's a bit tricky actually for environmental isolate if you use conventional biochemical test. But for those who have experience and very skillful person, they simple for them. Okay, and then another test that we did, nitrate, urease, nitrate, lysine decarboxylase, and glucose glucose OF, oxidizer or fermentator, oxidation or fermentation glucose, right? And then next, what we did, PCR. Actually, you can straight away do PCR, but last time they prefer do biochemical test before they do PCR, because during that time PCR is quite expensive. It's not like now. So now you can simply use PCR instead of biochemical test. And then they start do also fingerprinting for molecular typing using PFGE. So nowadays we can use other method instead of PFGE, because PFGE is the old method and a bit tedious, and the apparatus, the consumable is quite expensive. You can do RAPD, you can do RFLP, you can do other method, you can do sequencing as well, right? So summary for my talk today, right? Meliodosis is endemic in the Southeast Asia and Northern Australia regions. Sporadic or disseminated meliodosis is reported in other countries, and most of the cases were acquired from endemic countries. The agent of meliodosis is Burkholderia pseudomallei, a soil saprophyte easily found and isolated from contaminated soil and water, especially during the rainy seasons and typhoon. People who engage in outdoor activities such as paddy field farmers, gardeners, and healthcare workers who process clinical specimens related to Burkholderia pseudomallei should wear proper protective attire and seek treatment if any incident happen. Serological investigation in detecting IgM and IgG helps patients detect acute phase and chronic meliodosis infection and also exposure to the disease. Rapid identification of Burkholderia pseudomallei may use molecular methods to shorten the diagnosis time, but bacteriological confirmation is still needed. Vaccine development is needed as it will protect children patients from getting severe meliodosis infection. So with that, I thank you.